welcome everybody. Thank you for braving the freshly arrived winter out there. My name is Longtin. I'm University Research Chair in Neurophysics. I'm the chair of the Physics Department here as well at the University of Ottawa. And I work at the Brain and Mind Institute. And I'm talking to you on behalf of Michael Schlossmacher, the director of the Brain and Mind, who unfortunately is held up in clinic right now. So he cannot attend tonight. So we have a very exciting program, we hope, for you on decoding brain circuits and moods and thoughts. And our first, we're going to split the job here between the three of us. So Jean-Claude Béïque will talk first, and Naud, and myself. Naud is a professor in the Faculty of Medicine, neuroscientist, molecular cellular systems neuroscientist. So take it away, Jean-Claude.
All right, thank you. Yeah, I think everybody hears me. So thank you all for coming. If in the back, is that okay? Yeah, you seem to be raising your hand. If I speak too low, just raise your hand and I'll try to speak up. So when we came up with the title a few weeks ago, it seemed like a pretty good fancy title that kind of captured pretty much everything, a lot of the research programs that are actually being carried out here in Ottawa in the University of Ottawa Brain and Mind Research Institute, and especially the folks in the Center for Neurodynamics. However, this morning when I was going through our talks, I kind of realized that for a 1-hour talk, this title is rather grandiose. Hopefully we're going to be able to give you some snippets of part of the parts of brain research that we're trying to do here. So I'll begin. I'm going to focus mainly on memory and mood, and in doing so, I'm going to give you a bit of a neuroscience 101 type of background to hopefully lay the ground for Richard Naud, who's going to follow me and really talk about perception. How does the brain perceive the outside world? And then Andre is going to come in and finish up with more sort of a bit higher level circuit network and kind of give you a bit of a sort of a 30,000 feet level of how we think that circuit dynamics underlie thoughts. So to begin, I want to talk to you about memory. And one, I want you to have some sort of an abstract understanding of why is it that we learn. So in a nutshell, the answer is that it's evolutionary advantage if you are able to learn. So why is this? So let's consider bacteria. So bacteria have been around for 3,000 million years. So clearly, they're still around. So clearly, they're doing something good. And I want to give you just a little example. So what do? Well, one thing they do, they make DNA. Everybody knows about DNA. And they can code for proteins. In this case, ATP synthase. What does ATP synthase do? It does ATP. ATP is found in every single living organism. Every single cell on this planet uses ATP. See ATP as a form of, it's like little batteries. Synthase just charge up little batteries and then you have energy, and then your cell could use it to do whatever it wants to or it needs to. So this also, the ATP synthase in a bacterial cell and in the one in your bodies are about the same, and it highlights a fundamental feature of evolution. When nature finds something that works, it doesn't reinvent it. It keeps on using it over and over and over and over again. So now, what does bacteria need to do to be able to survive? Well, no disrespect to bacteria, it's fairly simple what they have to do. They need to take up some nutrients and reproduce, and they have been doing this close to forever. Now, if you look at evolutionary perspective, obviously you have all these organisms, all these species that are doing things that are somewhat more complicated than what a bacteria needs to do. So if you look at this, it's easy to get a pretty good intuition that the relationship between these species and the environment are a bit more complex than what a bacteria needs to do. So what is learning? So well, learning essentially gives you the ability to interact with your environment in a better way. Environment is not stable, it's static. That's a given. So if you're able to interact with your environment in better ways, the odds are you're going to be able to survive longer. And it's easy also to imagine that as you grow through evolution, say for instance all the way to humans, they will have a learning advantage. They will have an evolutionary advantage if they're able to learn. So what does memory do? Well, memory gives you a catalog of behavioral responses to given changes in your environment. If you're able to do this properly, you'll survive. Hopefully, if you put your hand on a stove, well, you do it only once, right? So you're able to learn from your environment and optimize your behavior to it. Now, even more, at a more abstract level, communication is a form of learning. If you have complex social structures between groups of individuals, if they're able to learn a language and interact with one another in a meaningful way, well, they have a better chance of survival than their counterparts, or say a tribe that's competing for limited resources. Now, so that's, I think in a nutshell, that's how we perceived how we conceptualize how we need to learn. And now, and this is everything is summarized here. It's easy to imagine that if you have a predator, if you're able to learn and to escape behavior, well, you have a better chance at survival. Now, how do we study memory? So when we talk about memory, now I'm going to switch gear a little bit. I'm going to talk about HM. So HM is an infamous patient. He's by far the most famous patient in cognitive neuroscience. And so his name is actually Henry Molaison. We've known his name only at his death in 2008. Before that, he was known as HM. So HM, when he was about 27 years old, he suffered from intractable epilepsy. So they tried drugs and nothing worked. And as a last resort, when you have intractable epilepsy, which in this case was believed to be caused by a biking accident when he was about 7 years old, they removed, during a surgical procedure, they removed part of his temporal lobe, including the hippocampus. They have essentially a chunk of tissue that has been removed. And HM was studied by Brenda Milner, who is faculty right down the highway at McGill University. She's still faculty at McGill. She celebrated her 100th anniversary over the summer. And she studied HM for a number of years, and essentially was able to show that the hippocampus is required for short-term memory. So she would see HM every single day, and every single day she had to reintroduce herself to the patient. He did not remember. He did not remember her. So he could not store new information. However, he could draw a plan. He could make a plan of his childhood house. And so he had really good memory until the surgery. Now, so this in and of itself is interesting, but the truly remarkable feature that Brenda Milner discovered while studying HM is given by this simple experiment. So she had HM draw a star, but he had to draw by looking in a mirror. And you cannot see your hands, but you only see your hands through the mirror. It's a hard task. And so when you're doing it, you're making a whole bunch of errors. But if you do it over and over and over, you get gradually better at it. And so HM was getting better at it. So he had a memory, he had a procedural memory of what he was doing. Yet, whenever the next morning Brenda Milner would come and explain to the patient the experiment, well, she needed to explain it all over again. He could not remember how to do the experiment. However, he was getting better at it. And then this led to the idea that all memories are not alike. There's different types of memories that are stored in different parts of the brain. And this was a truly revolutionary idea then. So now the point I want to bring too is obviously if you want to study memory, there's a lot of reasons to study memory in humans. And we can do this in a number of ways. You could do this by neuropsychological approaches, like the one used by Brenda Milner. And also you can use MRI, fMRI, and types of imaging in humans. Now the truth of the matter is this is great, but these techniques are highly limited. And they're limited in resolution, in time resolution, they're limited in spatial resolution. And so here enters really, and this is what we're going to talk about a lot today, enters animal models. So you can do experiments in animals that you cannot do in humans. And a significant portion of our understanding of basic aspects such as synaptic transmission, brain function, synaptic plasticity, or memory, it actually comes from animals. And this is maybe a little bit discomforting for some of us, but our brain and the brain of a rat or a mouse are highly, highly similar. They have the same structures. Some of our structures are larger than theirs, but all in all, it's the same thing. Again, this concept that the brain, when evolution or the nature develops something, just uses it over and over to do the same task. So if we talk about neuroscience in general, well, we have to talk about these folks. So we have to talk about Santiago Ramón y Cajal, for many, who's the father of modern neuroscience. And you had Camilo Golgi. So both these folks, they won a Nobel Prize in 1903. Although they were mortal enemies, they hated one another. When they got on the podium, they didn't even shake hands, they didn't even talk to one another. Essentially, Santiago Ramón y Cajal used Camilo Golgi's technique, and they were both using the same techniques and getting roughly the same results, but they both had fundamentally different interpretation, and they went through the neuron versus the reticular theory, and for history, Ramón y Cajal was right, and he said that the brain is not a big blob, it's individual neurons that each and every one of them can do their individual things. And I'm just gonna walk you through a few of these pictures that are actually more than 120 years old now. And they're still, I mean, he was right on 120 years ago. And I'm just gonna take a few seconds to go through, because they are actually gorgeous. This is an example of a retina. You have a number of different cell types. Here is an example. These are Purkinje cells from cerebellum with massive dendritic arborization. Now, this is the infamous hippocampus. That is the region in the brain that, if you have it removed, you can't store new information. And then here you have cortex. And then here you have a layer 5 pyramidal neuron with these extensive dendrites. These are like antennas in all of their glories. Now, I want to give you a sense of the scale of how big these things are. So this is a brain. It's about 50. This is a human brain, about 15 centimeters in diameter. And then all of this here, this is white matter. White matter is essentially electrical wires. You have a whole bunch of brain regions that need to communicate with one another. It goes through electrical wires. But here on the outer rims, you have here, this is gray matter. Gray matter, these are individual cells. There's about 50 microns in space here. So a human hair is about 50 to 100 microns. Now, if you go even smaller, these are dendrites, these are antennas, and then these little protrusions are about 1 micron. These are individual synapses. A typical pyramidal cell has about 30,000 synapses. It receives, every single point in time, receive inputs from about 30,000 synapses. And that's the essence of information transfer between individual cells in the brain. Now let's delve a little bit deeper into different scales. And I hope you're able to I want to hear, the reason I'm putting this is I want you to get a sense of a fundamental problem in the brain, which has to do with real estate. These are individual pyramidal cells, and then let's focus on one. This is packed with cells. It's packed with, this is one dendrite here, and then you see these are individual synapses, all these protrusions. And then here, everything is color-coded. These are processes from other neurons. Dendrites, axons, and support cells, and everything is densely, densely packed. Now, how does synaptic transmission work? Well, this is just shows a little cartoon where you have, this is an axon that comes from a neuron that's far, far away. And then this little color-coded, these are little individual action potentials. And then they go, and then once this is another neuron, and then here there's a tiny cleft here. And then if you go in, an action potential comes, and these terminals are filled with little vesicles. These are filled with neurotransmitters. And really, and when there's an action potential, then you release this neurotransmitters in the cleft, and it goes and activates the other cell. So this is chemical in nature, but that's the essence of synaptic transmission. This is what happens in your brain at every single point in time. Now, we're going to showcase a little bit, not so much like give you an idea of how neuroscience, modern thinking about neuroscience, but we're going to showcase a little bit some of the work that's being done in this very building, and how do we try to study the brain. And this is one example. So here you have a pyramidal neuron. This is a drawing from Santiago Ramón y Cajal from 120 years ago. Now, let's fast forward 100 years. And this is a neuron. This is the same one, but we're putting electrodes in these cells, in these neurons, and we can fill them with a dye. And the major difference between this neuron and the neurons from Ramón y Cajal is that his were dead, they were fixed tissue, and these ones are alive. So we can put electrodes in there, and so we're putting electrodes inside living neurons, and then we can essentially ask, what is it that a single neuron do? What is the nature of the operation and the mathematical operation that they do? So if you focus, this is one dendrite. Remember, these are the antennas. And then you have individual synapses here. And the trick is we can activate, we just shine a laser on this particular synapse, and then we can record the electrical event that is induced at a single level, at a single synapse level. So there's 1 out of 30,000 synapses, we can stimulate 1 or 2 or 3. Now, this is like how we like to view a neuron. That is, a neuron are input-output machines. I know it's a complicated concept, but really there's no other way in thinking about neurons. So neurons, like, they have 30,000 synapses. They're constantly receiving inputs. They're integrating them over time and making a decision. So here what we're trying to do, so this is a nice cartoon, but now what happens in real life? Well, this is a real neuron. It has all these dendrites. And we try to study, there's a number of things that we like to study. So one thing, we study processing at individual synapse. So we like to think that every synapse is an individual computational unit. And you see here that we put in these optical tricks, we put in lasers, and then you see this little blip that lasts for about 50 milliseconds or so. That is the time at which a single synaptic event occurs. And you can see here sometimes these little blips, they represent neurotransmitter release. And you see, so sometimes you have failures and sometimes successes, or failures and success. And there's a dynamical feature to this, and that provides single synapse some processing features. Now, it seems simple enough, but this is giving us a real headache because we're trying to understand what is the nature of the information that is being transferred at single synapse. Now, we could do processing at levels of individual dendrites. How do we do this? Dendrites are complicated beasts. You can activate 5 synapses that are close by, 5 synapses that are distant, and then essentially what we're trying to do is mimic what is going on in the brain. So these types of experiments essentially are available. So what we do with these types of experiments, by collaboration with mathematicians such as Richard and Andre, and then we take these things and we transfer them in these types of cartoons where we're trying to infer like computational meaning. We're trying to infer mathematical meaning. For instance, here they could do Boolean logic, and, or, and not. They can segregate, they can amplify inputs, they do low-pass filter, they do a number of stuff, and we're only skimming the surface of what neurons do and how do they do this input-input transformation. Now, why do we care about this? Is that some of these things are regulated by a number of ion channels, a number of ion channels that are dysregulated in some diseases. For instance, we're working now on an autism mouse model, and some of these features, not all, but only a specific portion of these features is altered when you're missing or you have a small change in the expression of a small ion channel. That happens to be altered in autism. So this input-output machine is somewhat altered in very subtle ways in autism, and that's exactly what we're trying to do. Now, what I was telling you that neurons are input-output, they take all these things, and their output is they're making these decisions, then they're spiking. They're making action potential. And then, so you can see here, they're going to make action potential. Here they're continuous, or here they're mainly bursting, for instance. That's the infamous elusive neural code. What is it that these spiking means versus that one? That's a neural code, and Richard's going to talk to you more about this in a second. Now, Ramon y Cajal, in 1894, he had an hypothesis about synaptic plasticity. And don't read all of this, but essentially he says one can admit as very likely that mental activity will change something in part of the brain that's most utilized. Essentially, if you're thinking long about something, something's going to be changed. And that's going to be changed by a physical substrate in synapse. Synapses are going to change in some way, and that's going to be a way that the neuron actually will learn, will encode information or store information. And it's truly remarkable that he said this in 1894 because by and large, modern techniques are now showing that he was right. And I'm going to give you a quick example of this. This is one example where you have this is again one of those nice pyramidal cells with these big, nice dendrites. We can activate with these lasers these little synapses. And then we activate that particular synapse a lot. And then we just monitor it, what happens to it. Well, that synapse become bigger. You can't really see it here, but trust me. And then this is one example. You see how this synapse becomes bigger. So this is a manifestation of synaptic plasticity. So I want to bring back here the analogy of a computer. So when you're working on your computer, and say you're working on a Word document, and then you press save. And let's imagine that you're an engineer and you're trying to reverse engineer a computer. Maybe you're going to have a system or maybe a microscope that's going to be powerful enough to be able to go on the computer hard drive and see when you press save, something has physically changed on your hard drive. So in neuroscience, now we're at that level. We know that there's something that has changed, that memory causes a change in some of the features of synapses. In much the same way that you could see that, say, a hard drive has been physically altered when you press save following in a Word document. However, again, if you're trying to reverse engineer your computer, it's going to be very hard to see how, or to conceptualize how are these little changes on your hard drive actually gives rise to a Word document, a music, a particular song that's on your computer, or an image of some sort. And that's exactly where we're facing right now. The neuroscience is stuck in we're trying to, we see where synapses are changing, but how does that relate to a memory of a childhood? How does that reflect to, say, you getting better at playing ping pong? How does that reflect when you're thinking about your grandmother? However, it doesn't mean that there's no theories, and I'm just going to leave you with one of the leading theory about how we think the brain stores information. And it has to do with, the analogy has to do with your grandmother. There's been this old theory about there's cells in your brain that respond to particular features that you've encountered during your life, and that reflect a given memory. And colloquially, it was referred to as the grandmother cell. So now it turns out that it's probably not that. What happens is that you have what we call a Hebbian cell assembly. And let me walk you through it. It's not that complicated. Imagine there's a group of neurons that are all interconnected. Now, you present a particular stimulus, in this case a circle, right? And then you have, you have a number of cells that respond to this circle. Now you do this over and over and over again, and you form in these interconnected cells plasticity in much the same way that I showed you before. You make these things stronger, and then you just have essentially a Hebbian cell assembly. Now later on, when you see your grandmother, you don't need to have access to all of your grandmother in order to recognize her. You can see your grandmother in the dark. You can maybe smell her perfume. You can only maybe hear her voice, and this is grandma. So you only need partial aspect of a given feature in order for you to complete, this is called pattern completion, to complete the memory of your grandmother. And this is essentially what happens here, where you have partial, like for instance here, you have partial sense of a circle, and then you only partially invoke this Hebbian cell assembly, but because these synapses have been potentiated, then you complete the pattern. This is your grandmother. Now, it's hard to find support for this. However, there's a couple of years ago, there's a paper that was done in humans, and they were presenting a number of faces, faces of blonde individuals, and some cells were responding, other cells were not. And then you had some cells that were that did not respond to blonde person, to male, female, but only responded when that person was presented a picture of Jennifer Aniston. So giving sort of credence to the idea of you have these Hebbian cell assemblies. Okay, in the last 3 or 4 minutes, how am I doing on time? I want to bring you a little bit, related thing that we do in the lab right now, where we're interested in how mood regulates our decision-making abilities. So I don't know if many of you, those of you who have kids may have seen the movie Inside Out, which is the story of this little girl, and you go inside her brain, and essentially you have these characters. You have these 5 characters and represent five emotions. You have fear, sadness, joy, anger, and disgust. And essentially these emotions are in sort of constant competition. And depending on who wins, then that's going to ultimately influence her behavior. It turns out that this analogy, and Inside Out, it's well documented that they actually they consulted with neuroscientists when they were doing their movie, and it's actually not far from how we conceptualize the brain. In other words, all these emotions are embedded in distinct circuits in the brain, and we have a pretty good intuition that this is the case. Now, the trick now is to identify or to associate these different specific circuits to these actual emotions, and to see how they change decision-making in animals and ultimately in humans. And the reason why we want to do this is that there's a clear translational potential. For instance, so when you say for anxiety disorders, you come into a room, you may be anxious. So that means that some part of your brain that are encoding, for instance, something that is aversive may be upregulated and encoding for something that's aversive, whereas in reality it shouldn't be. So you have all these competitions between these different networks that are encoding these different emotions. Now, how is it that we can identify which circuit encodes these different emotions and how that will influence our daily activities? And this is what we're trying to do. It's a tricky endeavor, but here enters optogenetics. And I'm going to just briefly walk you through it. And essentially, it has to do with the idea of using light to control the activity of specific types of neurons in the brain. So this is example where you have this is blue light. You can activate, this is a neuron that has this optogenetic tag. So essentially you take a protein that is sensitive to light. Normally that protein is not found in the brain. If you shine light on the brain, nothing is going to happen. But you express in only specific cells. This is thanks to recent advances in genetics. We can put in these light-gated channels in only the cells that we want, and then we shine light. So essentially, and then when we shine light, we can control the activity of that particular neuron. So imagine here you have, this is color-coded. This is a rat brain, and every single color encodes different, shows different circuits. So it's really hard to study, but how do I study only the yellow one and not the blue one and not the red one and all the other ones? So optogenetics allows us to do precisely that. We're going to put the optogenetics in one circuit that we're interested in, not the others, and then shine light and see what happens to the animal. So how do we do this? Don't care about don't pay attention to the acronyms here, but this is just one example where we've labeled only one particular pathway or neural circuits that we thought was involved for aversion. And then this is an animal, it is a mouse, and you see this light cannula here. And so this mouse expresses this light-gated channel only in one particular network. And then here with as you can see here, there's a light cannula that will deliver light to that specific network while the animal is engaged in a particular task. And now, if we want to do this with mood, and we want to see how mood, or circuits we think encodes mood, alter decision-making, for mice work, it's a little bit tricky, because mice typically don't tell us how they feel. They don't tell us if they're depressed. They don't tell us if they're happy. So we have to play tricks. And one of the tricks that we do is we have them learn stuff. And then if they're, for instance, we have them learn in this particular case to associate a tone with a reward. So I'm just going to show you real quick. So pay attention to the jaw of the, in this case it's a mouse. So we're going to put a tone, right? It's going to be written here, tone, and the animal knows that it's going to get a reward. In this case, it's a water reward. The mouse is thirsty. It gets a tone. He knows it's going to get a reward, and then it's going to start to lick. And look at the licking behavior. So let's do this. He's chilling out, and then there's a tone, and he gets all excited because he knows he's going to get the reward, right? And then the reward gets there, and then he gets really excited. Okay? Now, and let's do the same thing. But in this case, we're going to present the tone, but just right after the tone, we're going to shine light, and we're going to activate only one circuit that we think is involved in aversion. And then so the animal is, what we think is going to forego its reward. He knows he's going to expect a reward, but it's as if he wants to go out, and he wants to eat a berry, but then he's seen an eagle, and then we're telling him, look, you've seen an eagle, so don't go out and eat the berry or else you're going to die. So, but he won't tell us, so the way that we do this, we teach him to expect a reward. Just at the time he's going to expect a reward, we're going to activate this neural pathway, and if we're right, then he's going to forego its reward, and he's not going to be licking. He's going to have a freezing behavior. So we're going to do the same thing, but then we're going to see here, you're going to see a whole bunch of blue lights. Whoops. No. Okay. That's bad. I don't have the video. You're going to have to trust me on this. So essentially, so essentially what we do is that you give the reward, you give the tone, and then the animal expects the reward, so he starts to lick. And then essentially the laser gets turned on, we're activating this pathway, and it completely freezes. And it's quite dramatic. And even some animals, so the animals, they expect to they completely stop, so they've foregone their reward. So we're using this type of thing, then we're fitting this in mathematical models to understand how animals are processing features from their environment in order to guide their decision-making features. So obviously you've seen that was my last slide. So that's all I want to talk to you about for now. Hopefully you've been able to get a sense of synaptic transmission and this neural code, and then Richard is going to take from here and delve in more details.
Video, apparently that worked on the live stream. I have no idea, I'm not an AV person, but you will have the link on our website after the week, and so you could actually check that out to see if Dr. Béïque was actually telling the truth or not.
Better work, eh? I have a video too. Did you try it when you created the new?
Let's try going up here. Oh, so sad. Okay. You know what?
We can show the real video too. Oh yeah.
Oh goodness! Now we've got. Okay. Yeah, I don't like when presenter view makes things. Sorry about that.
No.
Why don't you just go directly from his USB?
So, I wanted to talk about the neural code and the problem of neural coding, and where it comes. The way I think about this is, say you try to read someone's mind. There, you have the sci-fi experiment where you're reading someone's mind. Well, say you're able to put an electrode in someone's brain. You know that the activity of the cells in there are encoding the thoughts. And this is electrical activity. So if you put an electrode, you should be sensing this. What would you record? What would it be? Can you actually, from that signal, try to read someone's mind? Well, this is actually done routinely. So I have here a video that's been recorded by a neurosurgeon who's inserting electrodes in a patient to cure him for various reasons. And they have to go about and orient, and in doing so they record the electrical activity and plot this electrical activity to a speaker. So you can now hear the electricity. And the action potentials that Jean-Claude was mentioning appear in the speaker as little clicks. Now what you're going to see and hear is an electrode played with the speakers here, and you will see here, you see my little cursor. You see the electrical activity here, as Jean-Claude was showing in his previous slides, and this is just the waveform, so it's the recording of this action potential. And well, Chad was in the room, I think was participating. Where's Chad? Yeah, here's Chad. So this is this is your hand, right?
That's my hand.
Yeah.
And the guy next to me, Michael, recorded the video.
There you go. So as he's touching, you hear increase in chattering. This is the cells in his brain encoding his touch. What I want to bring your attention to is the fact that it's very noisy. It doesn't come as a very clear signal. And not only this, is that when he's not touching, we're also hearing a lot of chatter. So the brain is constantly active, and when it responds to a stimulation, it does so with a lot of noisiness. And that's part of the problem of neural coding. And the other aspect is, as if you were looking at a waveform, it changes all the time, and we don't know why. The second aspect of neural coding comes when you look at a lot of neurons at the same time. So what I'm going to show you here is a movie taken using the tools of genetics that allowed only a subtype of cells to fluoresce when they're active. So now, instead of hearing the activity of the cells, we will see them as little blips of light that will show. I'll pause it, start again, just to explain what you will see. So every square here, except for the first one, every square is one layer in the part of the cortex that's receiving the visual information of a mouse. Every little dot, and even many dots that we don't actually see, are nerve cells. And when they fluoresce a little bit more, when you see them a little bit brighter, it means they emitted an action potential. They're active. So every layer, every square is a layer, and you have different layers like this in the visual cortex. So you roughly see different neurons on every square, meaning that you see overall something like 2,000 neurons. And that's corresponding to half a millimeter of, by half a millimeter of cortex. And what you'll see in the first square is the visual input that the mouse is seeing. Somehow, all the visual input will be represented in the activity of those cells. That's all what the mouse is using to represent the world. And all this information is in this little flashing that we see. Well, I have no idea how to take these dots flashing and reconstruct the image. That will be reading the mouse mind. And right now, we're not able to do that. So that's the problem with neural coding. It's figuring out what cell means what when they're active. So why is neural coding important, other than this science fiction experiment? It's very important for research, trying to understand various aspects of the brain. Say there's a disease that's affecting a mechanism in the brain that's affecting how these neurons, these nerve cells are representing information. Well, it would be useful to know exactly how they're really representing information, to be able to quantify or know how this disease is affecting this representation of information. But also, more directly, understanding how the neurons are representing the world and how they're interacting with our body is the problem of brain-computer interface. So when you have a cochlear implant or when you have a retinal implant, the goal is to connect the outside world with your neurons. So a cochlear implant, you'll have electrical stimulation in your cochlea that will then reproduce the sounds that you were supposed to hear well. Or retinal implants, the same for the retina. Vision, there's a little camera that transfers to activities in the retina, neural activities in the retina. To send that message, you should translate it and make it in a way that's understandable by the neurons. For the other way around, you'd say you're not able to use your limbs, you're paralyzed, and you want to type. You'd want to have a brain-computer interface that reads your intention so that you're able to type on a computer. There are invasive and non-invasive techniques, and Adam Sachs is a neurosurgeon who's doing brain-computer interface, and this is a little cartoon showing how this works. You have someone that has a reader of activity, of neural activity, could be inside or outside the brain, and then you're trying to read that signal to type on a computer. And you have to interpret that signal. You have to understand what it meant. So the common denominator to all this is a dictionary, that is the neural code. A way to translate from neural activity to something meaningful, or from external world to the internal world. But we don't know absolutely nothing about the neural code. So perhaps one of the most important first entry in that book was done by Lord Adrian in 1926, where he did an experiment with connecting electrodes with a loudspeaker again to hear the activity of cells. And there's those stretch muscles, those stretch receptors in muscle that our neurons are able to know how much the muscle is stretched. But he was putting an electrode in those, in a neuron of a muscle, and he was looking at the activity of that neuron, and he looked at various weights that would be hanging, so stretching the muscle more or less. And he saw that if there was a little weight, you have just a few action potentials. If you added a bit more weight, you have more action potential, and so on, for large weight having larger rate of action potential emissions. So that means that we can read how much weight there is by looking at how many action potentials there are. So that's the firing rate. The firing rate, the number of these discharges, is actually encoding the weight. So you can count the number of action potentials here, and you know exactly, there's that much weight. So this is the analogy here would be that of a neuron making smoke signals, where you have someone else trying to read the number of puffs that are there, and that means more something. The problem with this that intuitively appears to me is this is very slow. It can't work. You can't have someone waiting for all these discharges to appear. And in fact, cells are emitting 5 action potential per second. And so if you have to, given the variability, if you have to count always roughly the 5 action potentials, it's very slow. And to illustrate that I want to make an experiment on you. I'm not an experimentalist. I'm just doing data analysis for a living and making theories of the brain. I'm always jealous of Jean-Claude who's doing the experiments, and when I'm giving a talk, it's my chance. So I'll ask you to, as fast as you can, say whether there's an animal. I'm going to show images, and tell me whether there's an animal in the picture. Okay? So you report this by clapping your hands. Okay, as soon as you see an animal, you clap. If you don't see an animal, you don't clap, but you have to be as fast as possible. Okay, you ready? Okay, let's go. Good. Wow, I typically have more people signaling animals for the rocks. You were pretty good, smarter than my students. So I can report this experiment has been done before, and the reaction time, so the amount of time it took you to react from the start of the image to the time where you clap your hands was half a second. Now, to recognize an animal, the information must first arrive to your eyes, then it activates a bunch of neurons in the retina, and it starts its long way to the visual cortex, which is at the end of the brain, on the other side of the brain. It takes, just in getting there, it takes 1/50th of a second, 20 milliseconds. And then you need to treat that information, because this early visual cortex is only able to treat small features of this image. You need to treat that information and realize there was an animal in there. So that's done by going down the visual pathway, and finally in this area you start to realize, oh, there's an animal. But now you have to remember that you were trying to recognize there was an animal, and that goes to the place of the brain that's encoding your current intention. Your current intention is comparing what you're seeing with what you're supposed to do and say, well, you should actually clap your hand. Then you go all the way out, creating the plan to move your hands and clap, and that overall, just in delays, there's about 200 milliseconds, so 1/5 of a second that goes right there. And then you have about 10 processing stages. So that leaves about 1/20th of a second per processing stage to do the, oh, I received some input, I'm going to communicate that to the next processing stage. That's extremely short. An action potential is of that order of magnitude, maybe 10 times faster. So how can it do that how can it actually encode the information by counting the number of action potentials, this rate coding that I was mentioning? It's not possible like this. Well, the solution is that you have many neurons and you share the load. So you have to consider that many neurons all receive a similar input, and they will all fire, and then it's not the number of action potential over a long window that encodes the information, but it's number of action potential throughout the population that encodes the information. So I try to illustrate that here. So if you have some type of input that changes in time, that's all given to a population of neurons, an ensemble of neurons. Each neuron would respond with action potential in time. Now, instead of counting the number of discharges per neuron, what you do is count the number of discharge per unit time across the population. And that's the ensemble firing rate. And this way, you react very quickly, because if there's you only need to count it's only about how short you can count action potentials, and that's going to tell you how fast you can respond. Now the nice thing is that it looks like that's the way neurons are reading out the signals from other from other cells, because when there's an action potential in one cell, it sends a depolarization, as Jean-Claude was mentioning. It gives a depolarization in the next cell that's very short-lived. It lasts for a few milliseconds. And then the response on that side encodes again the firing rate of the population before. So you have a readout that neurons are able to access of that ensemble firing rate. Now, this is all common, this is the entries of the neural code book, and I want to talk about what I'm contributing to that neural book and the research we're doing here. And I'm not the only one to think this way, but there could be some additional information in the shapes of those action potential and the particular way they look. And in fact, I say actually the action potential don't all come alike. Some of them are different from the others. And I illustrate that here. I say there's two types of action potentials that are being emitted by neurons. One that is coming alone, and another that comes in a relative burst. And given the mechanisms that the neurons have for emitting those two types of discharges, I would assume that they represent different types of information. So I say that this is a burst of action potential because it comes in a close succession, and this is a single spike. And those two are events. There are two types of events in the brain. There's those bursts and there's those single spikes. And they can form the basis of a different neural code, a different way of reading the activity. And if I look at the ensemble activity, instead of counting the number of action potentials, the firing times, per unit time through the population, I could count the number of events, and I get an event rate. I could also count the number of bursts. So this is a burst, this is one burst, this is one burst. There's no burst here. So you see that the time-dependent signal is different. This one is constant, and here it's increasing. I could also look at the burstiness. So if I see in a population an event, what's the probability that it is actually a burst? So I can look at a fraction of the number of bursts over the number of events, and that's the burstiness. And every moment of time, that's another signal that can be represented in the response of cells. And now, oh wow, so what does that mean? First, can neuron actually do that? Can they represent two different types of information that are moving independently in time? What do they mean? So that's really the subject of my research. First of all, we think that these two types of information are fundamental to understanding the brain. Mainly because if you look at the brain, there's all sorts of connections going through the cortex, as I was showing earlier. But mainly there are some going up a certain hierarchy and other coming down. So the information comes first in the visual cortex here, and it goes up with blue arrows through the visual treatment pathway, where you can finally recognize what's in the image. But then every one of those centers is sending connections down that somehow it's some summary of what happened. It's the internally generated information that's sent back down. But every center receives these two fundamentally different types of information. One that is sensory and the other that is internally generated. And in an experiment done by my colleagues in Berlin, it's a very strange experiment where a mouse is asked to report an electrical pulse that appeared in their cortex. And the mice are really good at doing this. They sense a very, very tiny electrical pulse. And they say, oh yeah, there was a pulse. And then we look at the response of a lot of cells. And we classify two types of cells. And in one cell type, we look at the burstiness in red and the event rate in blue. And the burstiness co-evolves in one population type, and in another population type, the burstiness in red and the event rate in blue are crossing each other. So they are in time, they're representing different type of information. And in fact, the burstiness was a very good reporter whether the animal missed the electrical pulse, or whether it managed to report the electrical pulse. Whereas the firing rate was a very good reporter of the external information, whether there was an actual electrical pulse or not. So our view is that we can improve the way we understand neural coding by looking at those different features of the activity. And my summary slide to explain this is that every neuron in the cortex, at least, is able to make a neural double entendre. So what I mean by this is not that they do any sexual innuendo, is really that they are constantly speaking two intertwined languages that you can separate. And that's fundamental to communicate at the same time the stimulus information, what you really saw, from what you are internally generating as an expectation. So for instance, in this nice image here where you have a clear image, so your senses are unambiguous. There are a bunch of dots here. The pixels is black and white. But what is it? Is it a rabbit? Is it a duck? Your perception fluctuates from one to the other. So internally, somewhere in your brain, it's saying sometimes, oh, it's actually a rabbit. Oh, it's actually a duck. And it's switching like this. And throughout the brain, this information, oh, it's actually a rabbit, is broadcasted. And every cell somehow has access to this. And what I'm saying here is potentially the neurons are able to encode those two sorts of information to sometimes communicate just the pixels, really, or whether it was a duck or a rabbit or a combination of the two. With this, I wanted to take the opportunity to mention a few books that I found were good for me to inspire me on the subject of neurocoding and neuroscience. So I decided to bring them up here. Is a book on consciousness recently published by Stanislas Dehaene, and a book on mostly clinical studies, clinical tales by Oliver Sacks, is slightly older, but both really inspiring for me as a scientist. So I thought that I might share that with you. And for the more technical aficionados, if there are engineers in the room or physicists that want to know more about the brain, I co-authored a book on neuronal dynamics that is addressing people with a mathematical background to learn more about the brain and neurocoding. So with this, I will let André Longtin on stage.
Okay, thank you, Vishal. So I'm going to hold your attention for the next maybe 10, 15 minutes max. You'll get a sense of what it is to have an 80-minute class at the University of Ottawa. We have 80-minute classes here. So I'm interested in actual computations done by the brain. So I'm going to give you a few examples of work that we've done and others about trying to figure out exactly what might be going on to achieve certain cognitions. Okay? So there are many levels at which you can study the brain, as was already made clear by Jean-Claude and Vishal. You can study up here at the microscopic level, just individual synapses, cellular level, all the transfer of information. You can be looking at a population of cells, and the boxes come wide open now with this new observatory called optogenetics, where you can really observe many neurons at the same time. And that is like a new frontier completely. So single cells, population of cells, or you can look at huge portions of the brain, columns and whole areas lighting up. So there are questions you can address at all these levels. We're not the first ones to do this. So Avicenna, Arab philosopher from the 1500s, was very interested in neural circuits. So these were drawings from his book. So you had circuitry coming out of the eyes and out of the ears, and they had thoughts about how different areas of the brain were maybe processing these different inputs. Obviously, a lot of this work came probably illegally from exploration on cadavers. And same for Da Vinci. So these Da Vinci's notes. So you see all these different circuits. And it's very straightforward to come off of the sensory organs. Once you get deeper into the brain, it's a total mess. You don't know what's going in and what's going out. And in order to understand the neural code, we feel, like many others, it's a good idea to know what's going in and what might be coming out. I mean, what is the thing doing? If you start really deep into the brain, you don't really know what's going in and out because everything is connected to everything else. Okay? Nevertheless, you can make some headway looking at the hippocampus, obviously, for memory. But in terms of cracking the code, we're working a lot on the senses. So I'm going to give you some examples from the senses. The idea that you can actually do modeling in the brain, cook up theories of what's going on, goes back, well, to those two fellows, hundreds of years ago, but also is kind of cast in stone by Hermann von Helmholtz. So have you ever heard of this person? He was a physicist, but he also did neuroscience. And have you ever heard of conservation of energy? So that's his law. It's the first law of thermodynamics. That's what he is most famous for. But he also was interested in things like, when you're looking here with your eyes, you can actually read off these letters very easily, right? So even though your attention is in the middle, you can go and read off things because your attention is it's called covert attention shifts. So he was very interested in that. He was interested in auditory processing. He wrote a whole book on musical consonance and dissonance. He's also the one who discovered that the nerve activity going from one place to another does not propagate at an infinite speed. It actually, a lot of people thought that back then in the 1800s. It actually propagates at about 20, 30 meters per second. He got into huge philosophical debates with his father, who happened to be a philosopher, and this made no sense to his father, because whenever you willed something, the action and the will happen at the same time. There should not be any delay because of slow propagating activity. So he kind of set a lot of this thrust in motion of trying to explain things with theories in the brain. So, physical laws of brain function, we're still looking for them. Richard and Jean-Claude have shown some of them about plasticity, what's going on, Lord Adrian experiments. So let me just put the question a bit broader here. So you've heard of MRI, so magnetic resonance imaging, or functional magnetic resonance imaging. You see these beautiful pictures of the brain in great spatial detail, and you can see which parts of the brain light up when you do something or don't do something. So for example, which part of the brain, what it's showing you is which part of the brain is consuming oxygen, right? So you can see what happens when you're looking, talking, counting, meditating. There's a group at the university here that studies what happens when you're meditating. But what's the connection to all the stuff we just heard in the last 50 minutes? The neural activity. It's one thing to know, okay, it happens here in these millions of cells, but what is actually going on in there? How do you count? So that's what I do for a living. I try and figure out what those circuits, what those laws are. And the analogy for those of you maybe with more physical backgrounds, like magnetism, a rock might be magnetic. it's a macroscopic property of the rock. Where does it come from? Well, it's because the rock is made up of atoms, and the atoms, certain atoms have spins that come in even odd numbers, and some of them have odd numbers, and the spins give you magnetism. So there's a microscopic detail, and there's a whole beautiful theory for this in quantum mechanics and electromagnetism and so on, and we know what's going on to a large extent. But for the brain, we're still kind of lost, and it's an exciting time. So we have this field called neurophysics. It comes in various flavors. Some people call it mathematical neuroscience, because my mathematical colleagues do more mathematics. Others call it computational neuroscience. At the one end of the spectrum is something called artificial intelligence, AI, deep learning, and so on. We're trying to figure out neural spiking, these electrical spikes, and the brain theory. And there's many different other flavors of neurophysics, such as understanding optical imaging, interfacing neurons to materials to do brain-computer interfaces, magnetic field measurements, electric field measurements, the brain volume measurements, PET scanners, so on and so forth. It's a highly interdisciplinary field, so my students work in this. They come from physics. They have to learn some neuroscience, but they have to learn these different areas of physics. Because neurons are nonlinear, you need to know this stuff. There's millions of neurons sometimes, so you need to know this field. And then you need to know some information theory and electricity and biophysics. And all this is part of biophysics. So that puts you on the map, what we're doing. And here's a very basic computation that neurons do. We know they do this, but it illustrates the explosion in complexity that happens. Okay? So neurons connect to one another, but they connect positively or negatively. One neuron will excite another one, or it will inhibit the other one. And this is super important. And luckily, there's only two major possibilities, but still, because we're pretty lost with this. So here's a neuron, and it's getting these inputs like Jean-Claude showed you on the dendrites. And if it gets an input from E1 on this, from this neuron up here, comes here and excites, it's going to connect, and you see the voltage on the cell here, which normally hangs around minus 70 millivolts. But all your neurons are polarized. The inside's negative. If it gets an input from this guy, it goes up. So the voltage goes up a bit. If it gets an input from this neuron, that brings it down. That's an inhibitory input. What if it gets this guy and this guy right at the same time? Well, then nothing happens because they cancel out. What if they come a little bit separated in time? Then it'll go up and go down. What if you have three inputs in a row coming down this guy here? The first one brings it up, the second one brings it up, and now it hits this magical threshold. Once it hits that threshold, a spike is going to happen. The neuron is like an all or none. Once you hit that threshold, a spike happens, and all the other neurons it's connected to hear about it. Those are the spikes, these action potentials we've been talking about. That's why it's nonlinear. There's a threshold. What if you have E2 and E3, E2 and E3 adding up? What if you have 30,000 of these inputs, and some of them are inhibitory, some of them are excitatory, and they come in at different times? You imagine just the explosion of possibilities that this neuron is actually trying to compute, right? But that's one neuron. It's not 10 to the, like, it's not like the 100 billion neurons we have in our brain. So it's a complete explosion in complexity that we have to deal with. That's one neuron. Okay, and one thing we like to think about is, okay, this firing rate that BHAW was talking about, how many spikes per second the neuron is doing as a function of input. Well, sometimes it's a straight line, and that's nice. But if there's another neuron that comes and inhibits it, that straight line moves over to higher inputs, and that's called subtraction. Other kinds of neurons might actually cause the slope to change. That's called division, also called gain control. Sometimes it has these other shapes, and we're looking for basic mathematical functions that the neurons are doing, subtracting, dividing, or some mix of the two. We know they do this. This is one circuit we found on how it actually divides. That's a complete mess, right? It's not easy, but you basically have here's an experimental data recorded down the hall here, where you do a lesion to the brain, and you can go from this curve to this curve, and you know that that part of the brain was actually controlling and adding and making this divisive. How does it work? The sensory input goes straight to the cells, and then it goes up through here and comes back here, and somehow this causes the division. But we analyze that in detail to find out what's going on. That's how the cell divides. When this particular input comes in, it could change the rate at which it can change that slope of firing rate versus input. So, but it's a complicated mechanism, but that's the way life is. So we just have to accept that. The feedback we're talking about is this thing where, you know, the outside world comes in, the physical stimuli, they go to these primary receptors like your retina, they go in and they go up and they go up like these pathways that you've just seen. But you see that there's arrows going the other way, because there's actually stuff from higher brain that comes back and back down to here. It rarely projects back out to the receptors. But it means that if you're trying to understand what's going on here, you can see things coming in and how it affects it, but it's also affected by higher brain. Actually, most of the effect is from higher brain. Most of the activity is dictated by what you're expecting rather than what you're actually seeing. Why is this important? So neurofeedback, we know that comes in different flavors. It's responsible for switching states of the brain. We know that negative feedback can cause brain rhythms. We know that it helps you focus attention. We know that it can help you estimate some stimulus coming at you, and it helps in learning. We know all these roles. Why is it important? Why understanding feedback is important? Well, here's an experiment, with hearing implants. So you know that if your cochlea is defective, you can bypass it using a stimulator, right? It's going to go and stimulate the acoustic nerve. So the damaged cochlea can be bypassed by stimulating the acoustic nerve. That's a feed-forward strategy. The problem is you're still not doing too well at cocktail parties. So you're kind of confused by lots of different inputs. But what if the next station, so there's a lot of work and a lot of operations that cure this to some extent. But what if you get a tumor down the road from there? Down the road, there's a lot of feedback happening. Down the road, so here you can bypass the receptors, they're defective. But what if this part is damaged? You need to know what comes in, but you also need to know what comes from higher brain. That's why we need to understand the feedback here. We need to understand exactly how to fix that part. We don't quite know how to do that yet. So we're looking for the rules of feedback. Okay, feedback, circuitry gets it gets pretty intense, and we try to dissect it out and figure out what's going on using various experiments. These experiments done with these animals that have very simplified auditory systems, but they use electric fields instead of sound. So it's like they're tuning forks. They're like swimming tuning forks. So super simple auditory systems, one tone at a time. So one fish next to another fish, they feel each other's tone, and like two tuning forks that are not exactly the same frequency, you're going to hear the whoa, 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 and you're going to hear the beat. So we know how the brain processes the beat in these animals. And that gives information on how we process beats as humans. What if a third creature appears that he's at another frequency? Then the beat gets more complicated. It turns into this, right? It's going to turn into a more complicated waveform. And so we figured out the circuitry to go and extract just that waveform. We know that waveform, actually, there's like a very rapid part and there's a very slow part. And to make a long story short, we know that the input can go through an interneuron and through a slow synapse onto this cell, or it can go directly. So the fast stuff goes here and the slow stuff goes there, and it's like an AM radio. It just can extract the envelope. And that's very important for us to recognize sounds and speech and so on. So that's some of the circuitry mapped out for that case. frogs count. They sit in the swamp, they listen to the dozens of other frogs around, and they know who to mate with and who to avoid. How do they do that? They count the calls. Different species, different call rates, different pauses between the calls. Okay? And we know, and this is work done with BHAW, and the experiments are done by a colleague of ours in Utah. Here's the calls, the acoustic waves coming in, and this is what a neuron does. And if there's like 8 calls in a row, this neuron generates a spike. So there's a neuron in the frog's brain that says, aha, there's an 8 call coming down from that direction. I should go there. Okay, but now, if the call sequence is not quite perfect, if it's interrupted, this is not quite the perfect male, maybe, right? There's a little call interrupted here. Then it goes, boop, 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 and then the voltage goes back down. And then the next 4 come in, and it's not enough to generate this big spike. Too bad. Okay? So that neuron won't detect that 8 call, because the 8 sequence is imperfect. So we figured out how that counting circuit probably works. It's a mess. There's inputs coming in from the ears. It goes to these circuits with inhibition, excitation, and so on and so forth. But by analyzing it, we can figure out, okay, frogs have a counting sense. That book that Richard just showed by Stanislas Dehaene on consciousness, he's a big proponent for humans having this counting sense. So this is like maybe the guts of the circuitry for the counting sense. Now, the big questions we're all interested in at some point, some of us anyways, where does consciousness come from? So I'm going to go off the deep end here just in the last few minutes. So it's a source of controversy, especially for the brain-mind problem. Dualists think there's a different kind of theory for the mind part than for the brain part. And we tend to work with under the opinion that the brain activity can be explained by brain states and neural activity, and we go from there. So we look for physical correlates of consciousness, and the brain-mind problem, has a long history. Some people say, classical physics is enough. This is a very reductionistic approach. It's like particle physics, just everything can be explained in terms of little details, even consciousness. We're not there yet in any way. Some people think quantum physics is necessary. Some people think that all of physics is not enough anyways. There's always going to be something missing, no matter what you do. And are we machines in any way? Are we predictable? But all these things are kind of attached to what you've been hearing, the noisy brains. And so my view is, the brain really is an emergent computer. It puts lots of different elements together into bigger circuits and bigger circuits. And at some point you get what we are, but how to understand that is very difficult because we're scaling a lot of things, putting a lot of things together. And when you put little simple building blocks together, you lose track very quickly of what's going on. And in fact, there are things that one can argue that we just do not know all the physics. Because by putting a lot of these circuits together, I can't even predict what's going to happen if you put a lot of these frog counting circuits together. I just can't do it in my head. I can do that on a computer. Right? Schrödinger, famous Schrödinger equation, quantum mechanics, he wrote about this in the 40s. He said, what is life? And that's his book. And he has a section on determinism and free will at the end. And he doesn't think that quantum mechanics is very relevant to biology. And he thought the activity of the mind is based on what are called statistico-deterministic space-time events. It's basically what we've been talking to you about. Okay. And, but there's these equations that we didn't show you that underlie these spikes. They are new physical law. And there's probably a lot of other new physical laws we just haven't discovered yet. Turing, you may have heard about this guy. So anybody's seen the movie The Imitation Game? So he thought about patterns in nonlinear systems a long time ago, and he said by the year 2000, computers with 1 billion bits of storage should be around, and on average, an interrogator could be fooled 70% of the time talking 5 minutes to a computer. That was true around 2000. If you called Bell Canada, you know, you could. Okay, so, and he thought a machine cannot do anything new. But what about a machine with randomness? Well, that's a different story, and we are quite random. Our brain is extremely random. So it's an open problem. Once you know the coding mechanisms, you can figure out how to repair. That's the whole idea behind healing and why we're doing it here in the Faculty of Medicine. Okay, so with that, we'll conclude, and then question and answer period here, and also we can take it outside next to the food. Thank you. So do we have any questions now from the group? Yes.
So is the end goal of this maybe not 10 years, but 50 or 100 years out? You can actually model a brain well enough that you can say, this brain with these inputs will produce those outputs. Predict behavior.
Yeah, so I repeat the question. Is the idea like in 50 years from now we would have a good enough model that knowing the inputs, we would be able to predict the outputs of the brain? That's our hope. We think there's something to model that's, modeling is about prediction. Once you have it, once you have a model that's good, it could predict. But the thing is, the predictions are always going to be statistical because the brain is a statistical machine. You stimulate the same neuron 10 times, it'll do 10 different things. On average, it's doing about the same thing, but the 10 responses are different. And if you ask me the same question 10 times, I'm probably not going to answer the exact same way 10 times. I might not answer after the 8th time. Sounds good.
The batteries are gone. Okay. I didn't hear anything in your address about learning and plasticity, so that if you overlearn, a cell changes into something else. What happens to the memory that you're trying to store in those cells? Does it go someplace else? Does the neural pathway change? Some work done by Everett at Cambridge.
Who did you say?
Everett is in the field of addiction treatment.
I'm not aware of that. So, I mean, this is, it's funny because I was teaching about, does this work? No? Yeah? Does it work? It's funny, I was teaching about synapse today in a class with the students, and we were talking exactly about that. So, there's work actually done by colleagues of Richard and Andre, where, no, I think it works now. Yeah. Where people are really interested in figuring out how does what we call bounded synapse. This is what we've shown. Synapses are bounded, right? And then you allow them to fluctuate between states, plasticity states, and how does that will influence the memory storage of the system. It turns out that the memory storage is not infinite. It's finite. Right? If you have synapses that are in models, if you educate the models based on the type of experiments that we do, there are only a number of transition states that they can have. And because if you assume that they have a finite memory storage, then there's a trade that you have with the ability of your systems to be plastic and the ability of your system to store information. In other words, if your synapses are really, really, really plastic, they're gonna learn a lot. But then you're going to reach that maximum capacity fairly fast. So in other words, you need your synapse plasticity. It can't happen all the time. There needs to be what we call contingencies. In order for a synapse to actually show plasticity, it cannot happen all the time. It needs to be fairly hard for you to actually play with the strength of a synapse. And experiments tells us that all the time. You need to meet a number of contingencies for you to actually induce a form of plasticity. There's several types of contingencies that you need to meet. For instance, I could tell you my phone number once. Chances are maybe some of you will remember it, but chances are most of you won't. If I remember, if I recall, if I repeat my phone number 7, 8 times, then you're maybe going to be able to remember me. So that's one of the contingency. Another contingency is what neuromodulators do, for instance, norepinephrine. If I stress you out, your brain is going to be flooded with norepinephrine. That's going to change your contingency for synaptic plasticity. Most of you will remember exactly what they did on September 11th in 2001. Because at that point, your brain was flooded with norepinephrine. So that was one of the contingencies, that you've induced synaptic plasticity in some synapses that were involved in the hippocampus. They were flooded by norepinephrine, so you have these synapses. A number of synapses are coincidence detectors. They detect coincidences. In that case, it was something that happened, and the other coincidence was your brain is flooded with norepinephrine. Now, and I'm going to close on this, for the idea of addiction, and this is where I'm going to close the loop with addiction and plasticity. Like, the current models of addiction is that we know that there are certain neurons in the brain that encodes reward. Right? So they will encode reward. Anything that and again, it's for evolutionary perspective. You need to engage in events that are in behaviors that are rewarding. For instance, we would like to have chocolate. Sex is one. So you need to be able to do this. What do drugs of addiction do? They are believed to induce memory or abnormal plasticity in those networks that are involved in reward processing. That's what essentially cocaine and nicotine too will induce, the types of plasticities that I showed you, but in those circuits that are involved in reward. And then, so what people are trying to understand, how this plasticity in those reward circuits can be modulated to deal with drug of addiction.
What about learning through gambling?
I haven't yet. So, that's a good question.
Cocaine, the epinephrine?
So as far as, look, we haven't figured out yet. I haven't seen a mouse that likes gambling. However, what I'm telling you about, like all these different drugs, I can tell you that the abnormal plasticity in these reward circuits, every single drug of abuse does this. Heroin, cocaine, morphine will do it. LSD will not, and that makes sense because LSD is not addictive. So I would presume that gambling would do the same thing.
Just to comment on the first gentleman's, sort of draw an analogy of the larger the database, like when you continue to do your research, the more information you accumulate, the larger the database, and the larger the database, the more accurate your predictions will be from that database. And to do with the neuroplasticity and in addiction, it usually has to do with brain chemicals, like you say, the reward system of the brain, I believe, and it's mitigation of pain, and the neuroplasticity of those impulses that continue to reinforce that same synaptic pathway become the norm, and then you basically have to break out of that loop and interject some new information in order to make any kind of a change. But yeah, just a few thoughts that were rattling around up there.
But actually, you've nailed it. This is exactly how we see addiction. Is that, I showed you at one point there was a number of brain pathways that were all color coded. They all do different kind of things. It's like this inside out analogy is not bad, where you have brain circuits that are receiving inputs that are encoding for reward, but they're getting a whole bunch of inputs, and the brain does this all the time. It's hard, and actually Richard brought the point about the speed, but this is a good example. So we were talking about norepinephrine, and it's a little bit analogous to the point you're making about reward. So you have norepinephrine cells. They're embedded deep in your brainstem, and for instance, these experiments have been done in the 1970s. You record the activity of a norepinephrine cell, And then you show of a cat, and you show a flower, nothing happens. If you show a dog, norepinephrine is going to go up. It's going to go to these neurons are going to spike to the roof, and they're going to flood the brain with norepinephrine. But then it's a question is how does the animal know that this is a dog? And the pathway that actually Andre and Richard showed you, this is exactly what the animal needs to do. So he sees something that barks, that's fluffy, it's grayish, it has some sort of, and then the animal needs to say, oh, it needs to bind all these features together and say, oh, wait a second, this is a dog. And then he needs to, well, then somehow it needs to be processed by some certain centers in the brain that knows, oh, you know what, I'm a cat, I do not like dogs. And then make this association, and then send some information that are going to be processed deep down in these cells. These norepinephrine cells are going to sense this and say, oh, this is, other parts of my brain is telling me this is a dog. I don't like dogs. I'm putting all these contingencies together. I'm doing all these, and then I'm flooding the brain with norepinephrine. And then the infamous fight or flight response. Am I staying here and fight the poodle, or I'm out of here because it's a Rottweiler? Right? But you all need to do this, and so all these circuits come together in dynamical ways, and then, you know, as Andre said, sometimes you put 1 + 1, then okay, that makes sense, and then we could, a third feature of the circuit comes together, and then it gets a bit complicated, and then when you only deal with 4, then your brain explodes, or at least ours.
I'm going to use myself as an example of addiction. I'd smoked from before I was 16 until I was 50. When I was 50, I had bacterial meningitis, and I never wanted to smoke again. There were occasions when the action, like being a tourist, I always wanted to stop that cigarette, it gave me a pause, but I've never wanted to smoke again. And I used to jokingly, based on grade 12 science, say, well, I think that synapse killed one of the brain cells. Is that possible?
That's a hard question. Okay, it's a hard question, but it's grounded in something that makes sense. So nicotine. So why is it so addictive? Part of the reason why nicotine is addictive is that it has to do with so when you need to associate something with a response. Now, nicotine, when you take a puff of nicotine, it takes 7 seconds for nicotine to go to your brain. And it takes 7 seconds to go to those cells that I was talking about. Well, they're dopamine cells. And these are the ones that are involved in your reward circuit. So for you, it's very easy to associate a cue, in this case, putting a cigarette to your mouth, and something that comes. If the buzz of nicotine you would get would happen 2 days after, I mean, of course, you would not be addicted to nicotine, right? So the brain needs to associate this, and this happens through synaptic plasticity on dopamine cells. Now, dopamine cells receive a ton of inputs, including from a number of other regions, including one of the regions that I put in, there's an acronym in there that I didn't talk to you, but it's called the habenula. It's a region that encodes for bad stuff. So for instance, if you take a monkey and you train him to have a reward, and then you don't give him a reward when there's a cue, his habenula is going to go up. If you're worried, your habenula is going to go up. If I punish you, your habenula level is going to go up. But your habenula has a really, really strong input to those dopamine cells. So maybe when you had your episode of meningitis, and I want to relate to you to that level, but I've had bad oysters once, and I think people can relate to this, right? And then once you have bad oysters, then when you look at an oyster after, you kind of, I don't want that. So probably that there's some inputs, it's like the Inside Out movie, there's some circuits that are encoding aversion. And they're going to these dopamine cells that are coding for reward. And these things are in constant competition with one another. So it's not completely crazy to think that as you put it, that some synapses that are encoding from aversion, they killed it for you. Right? They killed this you seeking nicotine buzz to satisfy that reward center.
Okay, we'll take one more question, and then our speakers have generously allocated a little bit of time afterwards if you want to go back in the atrium there to ask them some questions. But for those who do want to head out, we'll have one more question and then head out into the atrium.
Hi, so you've done a lot of analogy with computer science and brain, and saying that the brain is kind of this random computer, and I imagine it kind of makes sense since like the computer science was created like based on like the model that we have of the brain. And nowadays I know that we talk a lot about machine learning, deep neural networks and things like that, and I was wondering what's your opinion on how this field of computer science will help actually understand the whole, like the brain that created it, kind of.
That's a fantastic question.
That's a fantastic question. And I'd like to answer that question in relation to previous comment regarding the amount of data that we now have. So, at the Allen Institute, their mission, it's in Seattle, it's an institute that was built by Paul Allen. And their mission is to understand the brain, but the way they approach that is accumulate data. So they have, a bit like the second movie I was showing, mice that are watching movies and they record from different cells in their brains, but they do that every day, 5 days a week, different cells, different animals, different movies, and they've been doing that for a good 10 years now. So the amount of data we now have is humongous. And I'm often there deliberating with them how to make sense of that data. And so we're now limited in the model that could then try to make sense of that data, could explain this. All our hypotheses fall short. The hypotheses of the neuroscientists of the 50s, of the 80s, they only explain about 1% of the activity we see in this data. And we don't really know what to do. And so some of the neuroscientists have turned to artificial intelligence, and even people there in the Allen Institute. And so they ask the artificial intelligence, well, make sense of that data for me. And they find that the artificial intelligence is better by a good margin to explain the data, the complexity of the responses. But now we're facing a different challenge. What does that mean? And right now, this understanding is still limited. What did the artificial intelligence understood that we didn't? Yeah, that's the current state of that aspect of research.
So maybe just
Clearly the models are, even deep learning is based on extremely simplified models of neurons that were hatched out in the 50s and 60s. So if we were to put any of the known complexity of neurons we've learned since then, like the bursting and Shah's talking about, the algorithms are probably going to be even more sophisticated and powerful. We may still not be able to understand them. but it's the computer science model, of course, evolved out of zeros and ones from neurons. Yeah.
So just a quick comment. Sometimes I feel like when we're having these public events is especially guys like us more than other folks, sometimes we end up, when we're having you guys are asking these questions, we all often respond, oh God, this is hard, we don't know. And there's a sense of a little bit sometimes of, it's a hard problem. And I don't want to give the impression that, I mean, neuroscience in the last, it has dramatically exploded. But the analogy with rocket science is a good one. In 1960s, JFK said, by the end of the decade, we're going to have gone to the moon. Right? So essentially what they had to do is build a rocket. Neuroscience right now, there's more money in neuroscience currently in the world than, or even in the US, like with the Obama Brain Project, there's more money into brain science than the entire Apollo project. So is it more complicated than rocket science? The answer is yes. It is more complicated. And our level of understanding is rising exponentially, but it's a tough beast. We're trying to reverse engineer something that is really, really complicated, and the only way that we're going to get at it is by, or at least we hope, is by when you have multiple fields that collide, when you have optics and electrophysiology and physics and math and computational neuroscience. When you put all this together, then you can start to make sense out of this complexity.